3.1 million children around the world die annually due to malnutrition, which accounts for 45% of all deaths under 5 years of age. While this might seem like a problem that should be addressed by politicians and economists, scientists are also at work trying to alleviate the effects of malnutrition in infants. Malnutrition in infancy is not only a short-term problem but also a long-term problem because it stunts infant growth, particularly height, which is an example of gene-by-environment interaction because malnourished infants are unable to grow as tall as infants that are healthy. Scientists specifically looked at Malawian infants with severely stunted growth and compared them to healthy infants. Since breastfeeding is highly recommended for infants and common in this area, scientists examined the breast milk from the mother of an infant with stunted growth and one with healthy growth in the study titled Sialylated Milk Oligosaccharides Promote Microbiota-Dependent Growth in Models of Infant Undernutrition, conducted by Charbonneau et al. They examined the molecular makeup of breast milk and saw that the healthy infant's mother's milk contained higher levels of human milk oligosaccharides, or HMOs, while that of the stunted infant's mother had lower levels. HMOs, which are carbohydrates, one of the 4 types of macromolecules, are known to support resistance to pathogens, gut maturation, immune function, and cognitive development. However, the effect of HMOs on phenotypic growth is unknown, so experimenters decided to focus on a particular type of HMOs called sialylated HMOs, or SHMOs. In a previous animal study in which learning and memory increased in piglets when sialylated casein glycomacropeptide was used to supplement sow milk replacer, sialylated appears to contribute to superior development and high intelligence. These stunted infants exhibited characteristics such as malformed ligaments, intestinal infections, and impaired brain development. Additionally, experimenters performed genetic sequencing on the genomes of a few of the bacterial strains that were present in the 25-member culture obtained from the feces of the stunted infants. Researchers hypothesized that the lower levels of SHMOs negatively affected infant growth. However, collecting SHMOs for the study would be difficult, so they used sialylated bovine milk oligosaccharides, SBMOs, which are structurally similar to SHMOs. Researchers fed 3 groups of germ-free mice a typical Malawian diet and SBMOs. The first group only received these 2 things and acted as a negative control. The second group was additionally given microbiota taken from the feces of a stunted infant. The third group acted as a positive control and was given inulin, which is commonly added to baby formulas. The process was repeated in another mammalian organism. Piglets were chosen due to their similar intestinal makeup to humans. Looking at the results, the experimenters found that the negative and positive control groups had no growth effects, which was measured by whether or not there was a change in body weight and lean body mass gain. However, the group of mice and the piglets that were also fed microbiota from the feces of stunted infants did exhibit promoted growth. This demonstrates that there is a causal, microbiota-dependent relationship between SBMOs and growth promotion. In particular, SBMOs allow substrates to properly bind to enzymes, which contributes to some extent to the observed growth promotion, though additional research is needed to better understand exactly how sialylated milk oligosaccharides accomplish this observed growth. Thus, this study has paved the way for a new feasible treatment of severe infant stunting. Even through this one study, scientists have already taken a step in the direction of discovering a way to alleviate the effects of malnutrition in infants. However, SHMOs is only one of many components in human breast milk, leaving a lot of research that remains on the many other components and their effects on infant growth. With this continued research, perhaps in the near future, scientists will be able to partner with companies and receive funding to actually implement this new feasible treatment for severe infant stunting in communities like those in Malawi. Though these global challenges have no simple solutions, they illustrate the importance of the collaboration from members of all disciplines.